While walking on a street, sometimes you come across a stray dog sitting under a concrete bench. Have you ever noticed what is the difference between these two? The bench and the dog? Well, the major difference between these two is that the concrete bench is a non-living object, whereas the dog is a living being, and the dog can perform certain functions or activities which the bench cannot. For example, growth, metabolism, reproduction, and consciousness. That is, dog has some amount of consciousness, but the concrete bench will not. So these functions are performed by different sets of organs. For example, lungs are present in our body which help us in respiration. Heart helps in circulation of blood in the body. Kidney helps in filtration of blood, and many more. So these organs are made up of tissues which are made up of cells, and thus cell is the basic structural unit of a organ. So now we'll study about its discovery of the cell. So the credit for this goes to the English scientist Robert Hooke. He assembled a simple microscope, and in 1965 he observed a very thin slice of cork under a primitive microscope. So the cells of the living organisms could be observed only after the discovery of an improved microscope. So very little was known about in the next 150 years, because with a simple microscope we will not be able to get much information. So then the cork was obtained from the outer covering of a tree, which is called the bark. So he observed that many little partitioned boxes or compartments in the cork slice. So these boxes appeared like honeycomb, and he termed these boxes as cells. And he also noticed that one box was separated from the other by a wall or a partition. So when Hooke observed as boxes or cells in the cork slice was actually the dead cells. What Robert Hooke saw are dead cells. So he got his work published in his book, which is called Micrographia. His work was published. So living cells were first of all observed by A.V. Leeuwenhoek, Antony van Leeuwenhoek. So Robert Hooke has observed the non-living cells, but Antony van Leeuwenhoek has observed the first living cell. And he observed bacteria, amoeba, sperms, and red blood cells. So living cells could not be observed only after the discovery of the improved microscopes. So with the help of their high magnification, we are able to see the living cells. So today we know a lot about cell structure and functions because of these improved microscopes, such as the electron microscope. So now we'll study about the cell in much more detail. Cell. A cell could be compared to a brick wall. A cell can be compared to a brick wall that serves as the basic structural unit of a building. So the buildings, though built of similar bricks, have different shapes and sizes. So similarly, in the living world, organisms may have different kinds of cells but the cells of the living organisms are complex living structures, while the bricks are dead structures. So most of these cells are too small that we cannot see with the naked eye, but an ostrich egg is represented as the large single cell which could be seen with an unaided eye. So organisms show a variety in their cell number, shape and size. So most of these cells are very minute, and to clearly observe and study the cells we need two things. The first one is a device which magnifies the size of the cell. So the various magnifying instruments are used, such as magnifying glass, light microscope and electron microscope. And the next requirement is stains, that is special colors which are used for the various components of the cell. For example, methylene blue, saffron, etc. So now we'll study about the number of cells. So the number of cells found in an organism vary with the size of the organism. So the size of the organism is directly proportional to the number of cells present. That is, the elephant. In the elephant, the number of cells present is more in comparison to the number of cells present in the rat. So on the basis of the number of cells that form the body, living organisms can be of two types. That is unicellular organisms or multicellular organisms. Unicellular organisms are made up of single cell. A single cell organism performs all the necessary functions for survival, like taking the food, its breakdown, assimilation, respiration, excretion, growth, reproduction, etc. So some common examples of unicellular organisms are amoeba, paramecium, euglena, chlamydomonas, which are eukaryotic, and bacteria, which is prokaryotic. Multicellular. So multicellular organisms are made up of more than one cell. So they have more than one cell. And the number of cells may vary from a few cells, as in case of Spirogyra, to billions or trillions of cells, as in case of a banyan tree. So human body has trillions of cells, in which many different shapes and sizes are to be seen. So in multicellular organisms, different groups of specialized cells perform different functions, or there is a division of labor. So in multicellular organisms, we find the division of labor. But in unicellular organisms, there is no division of labor, because only one cell has to perform all the functions, in which the multicellular will have different groups of cells performing different functions. For example, in a plant, the green leaves produce food, while in the non-green plants present in the center of the stem help in transporting the food produced to other parts. So if we have a plant, the leaves produce food, while the stem will help to carry the food to different parts of the plant. So there we find the division of labor. Majority of the multicellular organisms begin their life as single cell, that is the fertilized egg or zygote. So even multicellular organisms, most of them start with a single cell, and this fertilized egg multiplies and the number of cells increases with course of growth and development of the organism. Now we'll study about the shape of cells. So a cell may have a particular shape as that of chlamydomonas, or it may have indefinite irregular shape as in the amoeba, that keeps on changing its shape by giving out projections of various lengths, which are called the pseudopodia. Pseudo means false and podia means feet, false feet. So these projections appear and disappear as amoeba moves or feeds. On the other hand, the definite shape in the chlamydomonas is due to the rigid cell wall and cell membrane covering the cell. So when we find amoeba, so if there is any food particle it has to engulf, then it immediately changes its shape according to the food particle and engulfs. So this is called the false feet or the pseudopodia. That is why amoeba has irregular cell shape. A white blood cell in the human blood is also a single cell which can change its shape like amoeba. But white blood cell is a cell, whereas amoeba is a full-fledged unicellular organism capable of independent existence. So even though WBC is also single cell and irregular in shape, but there's a difference, that is amoeba is a completely an individual entity which can live alone, but WBC is just a cell which is a part of the human body. So organisms that are made up of millions of cells have different kinds of cells such as blood cells, muscle cells and nerve cells, as in the case of human beings, with different shapes and sizes. The shape of the cell depends upon the function. So it is important to remember that shape depends upon the function which the cell performs. So generally the shapes are round, spherical or elongated. However, some cells are long and pointed at both the ends, exhibiting a spindle shape like smooth muscle cells. Others can be long and branched in case of nerve cells or neurons. So the nerve cell receives and transfers messages and helps to control and coordinate the working of different parts of the body. So the components of a cell that are enclosed in a membrane is called the cell membrane. So the components of the cell are enclosed in a membrane. So this membrane is called the cell membrane that provides a specific shape to the cell in both the plants and animals. But in plants, an additional covering is present over the cell membrane, which is called the cell wall. Cell wall is also present in the cells of bacteria and fungi as well. It gives shape and rigidity to these cells. So plants have an extra cell wall because they have to overcome the harsh environmental changes. Next, we'll be speaking about the size of cells. So size of cells in living organisms may be as small as millionth of a meter, that is micrometer or a micron, or maybe large as a few centimeters. So most of these cells are too small and not visible to the unaided eyes. So these could be seen with the help of a microscope. So the smallest cell is that of mycoplasma, which is a genus of bacteria, that is 0.1 to 0.5 micrometer in the diameter, and the largest cell is that egg of ostrich, that is 170 mm and 30 mm. So regarding the size of the cells, we can understand that the size of the cell has no relation with the size of the body of the plant or animal. The cells that form the leaf of a large banyan tree are not much different in size from the cells that form a leaf of a small wheat plant. And the size of these cells is related to its function. So even the size of the cell is also related to its function. For example, nerve cells both in elephant and rat are long and branched. So they perform the same function, that is transmitting long length messages. With this we have completed the size of the cells. Now we'll study about cell structure and function. So in multicellular organisms, several similar cells are grouped to perform a specific function. A group of similar cells performing a specific function is called a tissue. So the various types of tissues constitute an organ, and the various organs constitute to perform a specific function, and these organs together constitute the organ system, and the various organ systems constitute the entire organism. So right from the cell, the tissue, organ, and organ system and organism. So for example, the various tissues form a digestive system or a digestive organ like stomach. So first, there will be various tissues which form stomach. Okay, and along with many other tissues, there will be the formation of liver, intestine, and many more. And these all together constitute the digestive system. So each organ systems like the digestive system, reproductive system, respiratory system constitute the whole human being, that is the whole organism is found. So different organs of the higher plants are root, stem, leaves and flowers. So these organs perform specific functions, that is root helps in the absorption of water and minerals, and leaves are responsible for the synthesis of food, and stem is responsible for the transport of the prepared food. So in all these ways, the whole organism will be able to survive. So now we'll understand about the parts of the cell. So in spite of variation in the size, shape or form, every cell has a particular structure. So structure includes three basic components. The first one is plasma membrane, next cytoplasm, and the nucleus. Every cell should have these three basic components. So how to observe the basic components of the cell? So in order to observe the basic components of the cell, we can take a small inner shelly leaf of onion bulb and take out the peel from its inner side with the help of a forceps or with your hand, and now place this small piece of the thin onion peel in a drop of water on a glass slide. So the thin layer can be cut into smaller pieces with the help of a sharp blade before placing it on the glass slide. So add a drop of biological stain, for example methylene blue for animals or fungal cells and saffron solution for plant cells, into the layer and place a cover slip on it and ensure that there are no air bubbles. So observe this slide under the microscope and you will be able to observe the basic components of the cell. So it is important to remember that the stain used for animals and fungal cells is methylene blue, and for animals we use saffronin. So this is the activity which can be done to observe the basic components of a cell. Now we'll study about plasma membrane. Which is also called cell membrane. So it encloses the inner gel-like structure or the inner gel-like material which is called the protoplasm. That is, protoplasm is cytoplasm plus nucleus. So these are covered by a plasma membrane or cell membrane. So protoplasm is a living substance of the cell and it separates one cell from an other. and also form the surrounding medium. So it is thin, elastic and porous in nature, but it allows only some selected substances to pass through it. That is why it is called selectively permeable, and it is mainly formed of proteins and lipids. So the functions of plasma membrane are that it gives shape to the cell, and it permits the entry and the exit of selected materials in and out of the cell, and it maintains the individuality of a cell by keeping it separate from the adjacent cells and from the surrounding medium. Next, we'll move on to the cell wall. So cell wall is a rigid, supportive or a protective and fully permeable covering present outside the plasma membrane. This is the cell wall which is present outside the plasma membrane. In plants, it is present in plants, fungi and bacteria except in mycoplasma, and it gives definite shape, size and support to the cell. So the functions of cell wall are: it provides protection to the plasma membrane in the protoplasm, and it provides mechanical strength and support to the cells, and it is freely permeable to water, substances and solutions, and it protects the cell against pathogen and mechanical injury. And it is important to remember that cell wall is absent in animals. It is not present in animals. It could be seen by the observing the chick cells which show plasma membrane, nucleus and cytoplasm. We won't find cell wall in animals. Next, we'll be studying about cytoplasm. So it is a jelly-like substance present between the nucleus and the cell membrane. So it is a jelly-like substance which is present in between the plasma membrane and the nucleus, and it is formed of proteins, carbohydrates, lipids and certain inorganic substances including large amount of water. In the cytoplasm, many other components of the cells which are called the cell organelles are present. These are mitochondria, Golgi bodies and ribosomes. Now we'll study about plastids. So these are scattered in the cytoplasm of the leaf cells. So they are of different colors, and some of them contain green pigment called chlorophyll. And the green colored plastids are called the chloroplasts, and they provide green color to the leaves. And chlorophyll in the chloroplasts is essential for photosynthesis. So photosynthesis is the process by which plant prepare its own food. Next, vacuoles. So these are the single membrane bound spaces, and the vacuolar membrane is called the tonoplast. So this is a vacuole and its membrane is called tonoplast, and these are the kind of storage sacs. And in mature plant cells, vacuoles are large, central, whereas in animals they are small. So in plants they are large, while in animals they are small. And in mature plant cells, the vacuoles occupy a large portion of the cell volume. So with this you have completed cytoplasm. Now we'll move on to nucleus. So nucleus was first known by an English botanist which is Robert Brown in 1831. So nucleus is a major cellular component. It is generally spherical in shape and located at the center of the cell, but it may also occupy the peripheral positions. It can be stained and seen easily with the help of microscope, and it is bounded by double nuclear membrane which separates it from the cytoplasm, and its membrane is porous and allows the movement of materials between the cytoplasm and the nucleus. So the matrix present in the nucleus, so the matrix which is present in the nucleus is known as nucleoplasm. So this is the matrix which is present inside the nucleus, and the nucleus contains a small spherical body which is called nucleolus, which is the site of ribosome formation, and hence it is known as factory of ribosomes. After their synthesis, the ribosomes come out of the cytoplasm through the pores present in the nuclear membrane. In addition to nucleus, in addition to this factory of ribosomes, the nucleus contains chromatin fibers. So these fibers condense and form chromosomes. So the chromatin fibers will condense and form chromosomes during cell division. So these chromosomes basically carry the genes that help in the transfer of characters from the parents to the offsprings. So they can be seen only when the cell will divide, and it consists of two arms, that is two similar threads which are called the chromatids, and these chromatids are held together by a centromere. So the function of nucleus is it plays a major role in the inheritance of characters from one generation to another, and it controls all the activities of the cell that it performs during its life cycle, and therefore nucleus is called the brain of the cell. Next, with this you have completed nucleus. Now we'll study about the prokaryotic and eukaryotic cells. So based on the complexity of the cell structure, the cells are of two types. The first one is prokaryotic cells, and the second one is eukaryotic cells. So prokaryotic cells. Nucleus of the prokaryotic organism is not well organized, and there is no nuclear membrane, and the cells in which nuclear material is not enclosed by a nuclear membrane are called the prokaryotic cells, and the organisms consisting of prokaryotic cells are called prokaryotes. Pro means primitive and karyon means nucleus. So the region where genetic material lies in the prokaryotic cell is known as nucleoid. So the region where the genetic material is present is called the nucleoid. And the membrane bound cell organelles like mitochondria, Golgi bodies are absent in cells, but membraneless organelles like ribosomes are present in prokaryotes. So the examples of prokaryotes are bacteria, archaebacteria, mycoplasm, and blue-green algae, which is also called cyanobacteria. Now, eukaryotic cells. So cells like onion cells or the plant cheek cells have well-organized nucleus with double-layered nuclear membrane. And such cells which have well-organized nuclear membrane are called eukaryotic cells. Here U means true and karyon means nucleus. So these cells contain the membrane-bound organelles like mitochondria, Golgi body, and they also have ribosomes as well. Now we'll come to the comparison between plant and animal cells. So first, the cell wall. Cell wall is present in the plant but it is absent in the animal. Cell membrane is present in both plant and animal. Nucleus is present in both plant and in the animal, but in the plant it is found in the corner, while in the animals it is found in the center. And the nuclear membrane is present in both. Cytoplasm is present in both plant and animals, and plastids are present in plants but they are absent in animals. Vacuoles are present in both plants and animals, but in plants they are large while in animals they are small. And here, coming to the Golgi bodies, the plants have simple Golgi bodies which are called the dictyosomes, while animals have complex Golgi bodies, and centrosome is not present in plant cells while it is present in the animals. So to understand more about the cell organelles, we'll study about them in another video. With this, we have completed about the comparison between the plant and animal cells as well.